Hey 7th graders, if you guys are watching this, you guys are either working from home or I am gone today and you guys are working on your mutations assignment with a guest teacher. With that being said, I'm going to let you guys go ahead and work with your table partner or whoever you guys worked with on your DNA lab station if you guys are sitting in class. Obviously, if you guys are home, you guys are working on this by yourself. If you guys are working on it with a table partner, make sure you guys put you and your table partner's names right here on the first slide. Okay, again, this is the mutations assignment. In your learning targets, I can describe a mutation in DNA as either good or bad and explain why. And I can explain how a mutation might change the structure of DNA and proteins. Okay, so with that being said, let's go ahead and take a look at your guys' bell ringer today. So with your table partner, you guys are going to answer this question. Do you think mutations are good or bad? Circle your answer, okay? And then explain why you guys made that choice or choices. Okay, once you guys have that done, we're going to go ahead and move on to your introduction here. So here is your guys' link you guys will need for today. So you guys will right click and open that link up. And you guys will see this little simulation. Okay, with that being said, you will open the mutations link below. What you see on the screen is a normal piece of DNA without any mutations at all. As you know, DNA gives us the code that makes proteins, and proteins help build our body and make it function. So we talked a lot about your eye color, okay? DNA doesn't make your eyes brown, but DNA is a recipe that makes a certain protein, and that protein's physically what turns our eyes brown in color, okay? It also produces a lot of proteins that help our body function correctly. So as you know, again, DNA gives us the code that makes proteins, and the protein helps build our body and make it function. We're going to look at how mutations in our DNA change the protein we build in our cells and bodies. So I did kind of some of these for you guys so you can see examples. But we're going to go ahead and take a look at this simulation and look at a normal protein. So as you guys see it when you first open it up, this is a normal piece of DNA. Okay, what you guys will click on down here is show protein. And what you guys see happen is that protein is going to show you the amino acids in that little chain. This is similar to what you guys did in the DNA lab stations with the piece of pipe cleaner and the beads. Okay, so it kind of does this all for you guys. This would be kind of your guys' sequence of beads here. So you guys will go ahead and snip that. When you guys snip it, this will pause. Okay, and then you guys will screenshot your protein onto this page. I've already done that for you guys, so you don't have to do that one. Here's your example for you. So what we see is that normal DNA strand without any mutations is going to give us this protein that looks like this. So it's going to be met, proline, glycine, glutamic acid, serine, leucine, isoleucine, glycine, and leucine. Okay, so you guys can see what that little chain of amino acids looks like. Okay, what you guys will then do is on part 2, you guys will look at a couple different mutations, and you guys will see how those mutations have changed that protein. If you guys actually see a change, you guys will take your little highlighter and you guys will highlight where you guys see differences in the proteins. So now you will try 7 mutations to see what effects those mutations have on the proteins they're supposed to make in comparison with your normal protein. Generally, if a protein is different, it will also function differently. For this activity, and when I say it functions differently, maybe it functions worse or doesn't function at all. Maybe it functions better. Okay, so kind of refer back to that bell ringer you guys did. Maybe it doesn't change it at all. Okay, so it's going to have the same function. So for this activity, you will click on the 8th base pair on the top. In the normal DNA, it is an A. Try one of each mutation for the 8th base pair and click on show protein. Screenshot your proteins. Compare to the normal protein. Highlight where the protein may be different if there is a difference. Hit the restart button in the, I'll show you guys just the reset button between each mutation to reset it. So I did a couple examples for you, including this first one. So what I see right here is, here's my normal protein, okay? And the protein I'm looking, or the mutation I'm looking for is a substitution from A to T on that 8th protein. So let's take a look at what that would look like. So I'm gonna go ahead and hit reset. Here's my normal protein again, and then I'm gonna count the 8th base pair. So here's 1, 2, 3, 4, 5, 6, 7, 8. So this is the A that I was talking about that we see as the 8th base pair. Ugh. So I'm going to click on that. And my first one I'm going to do, if I look back, is a substitution. I'm going to change that A to a T. So here I am on my 8th. I'm going to go A to T. And I'm going to click then Show Protein. Okay? And then like I already did, I'm going to go ahead and snipping tool this protein. So it would look something like this. Again, I've already done this for you guys, so here's that picture for you guys. And then what you guys can do is you can compare. All right, here's my mutated protein. Here's my normal protein. My normal protein should be MET, PRO, GLY, GLU, SER, LU, ILE, GLY, LAU. Okay, so this protein that was mutated is exactly the same as my normal protein. So I'm going to go up my draw tool and I'm just going to write same. Okay, so you guys will do the same thing. Okay, then on slide 7, you guys will make this change yourself. You will screenshot it. And again, if you guys see any differences between the normal right here and what you guys see in your mutated protein right here, highlight where you see the differences. If it's the same, do just as we did on slide 6 and write the same. Okay, so on slide 7, you guys are going to do a substitution mutation. You're going to change the A to a G. So again, you guys will go right to reset. You'll click on that 8th protein, substitution, A to G, and then go show protein and take a screenshot of that. So there's my mutation. There's my show protein. Screenshot that, put it right here, highlight any changes you see. Slide 8, you're also going to do a substitution, but you're going to change the A to a C. Okay, again, highlight anywhere there might be different. If there's no differences, write same. Okay, on slide 9, I did this example for you guys. Well, you guys are going to insert and add an A. So let's take a look at what that looks like. I'm going to go mutated. I'm going to click on my 8. I'm going to go insert, and I'm going to insert again an A. So insertion, insert an A, and then show protein. Okay, and then I'm going to screenshot that. Again, I already did this for you guys, so you don't have to worry about screenshotting that. You guys will highlight everything that is different. So I already have my draw tool pulled up. Okay, and let's go ahead and compare. All right, I've got MET, PRO, and then look, this should be a glycine, this should be a glycine, glutamine, serine, leucine, so that's normal. This one's not. That should be a leucine. This should be an ILE. This should be a GLY. This one's an LEU, and then this one's an extra one on the end. So actually, out of all of this, only 4 amino acids are correct. So I've highlighted all the ones that are not correct. Again, if you guys come across any of these where you insert a letter and it's the same, you guys would just write same. Okay, so highlight what's different on this. Compare it to, again, your normal chain right down here and highlight where you guys see difference. Remember, you're always starting with methionine or MET. Okay, all right, next one, you guys are going to do the same thing. You're going to insert a T, so you guys would hit reset. You're going to go to this one, insert, and insert T, go to show protein, screenshot it, highlight where it's different. Okay, slide 11, insert a G, and slide 12, insert a C. Okay, and then your last little one that you guys are going to do in this simulation is a deletion. So you guys are going to remove that A. So you guys, again, will go to reset. You'll click on that 8th one, and we're going to just delete it. There's really only one change you guys can make with that deletion. Go ahead, click show protein, insert it right there, and highlight all of your amino acids that are different. Okay, once you guys are done with that, you guys are going to go on to the last 3 here. You guys have an extra credit option if you want as well. Okay, I suggest everyone do it because what does it hurt if you guys get an extra credit point? So below there is a picture of a normal segment of DNA, and then there's also going to be a mutated segment right underneath it. Compare the pictures in the questions with the normal DNA to determine whether there was a substitution, an insertion, or a deletion mutation that occurred and then where it's occurred at. So let's do the first one together. So really, you guys will only have 2 to do on your own plus the extra credit. So let's take a look. Here's my normal DNA. And then here's my mutation number 1. So here's mutation number 1. So I'm going to go ahead and compare. So I got CG, AT, TA. So it looks like there is a difference right here. If we look it looks like we've actually deleted, let me kind of erase these scribbles for you guys. Okay, we've actually deleted so this guy was here, and then we deleted it. So these two line up, these two line up, these two line up. So you guys can go through and see how everything else is the same, except for this one base pair that it looks like we deleted out of the normal. So you guys would go ahead and circle, all right, this was a deletion. And which one did we delete? So look at the normal. 1, 2, 3. We deleted the third one. Okay? And then describe your change. So we deleted the AT was deleted. Okay? So that's how you guys would describe the change. Okay? You guys are going to do the same thing. Figure out what's different about mutation number 2. Tell me where and describe it. For this one, again, you're going to figure out what's different between the normal and mutation number 3. Tell me if it's substitution, insertion, or deletion. Tell me where and describe the change. And then finally, this last one, extra credit, is a challenge. So you guys have 2 mutations that occurred. So you guys are going to want to do 2 different colors for this. So for the first one, do a circle using one color, what mutation you saw and where, and then change your color and circle the second type of mutation, either substitution, insertion, deletion, and then tell me where, and then describe your 2 changes. If you get it correct, I'll give you the extra credit point. So I do want everyone to do this. It's a challenge. If you get it right, great, you guys get the point. If not, you guys still get 100% if you did the whole assignment. Okay? Other than that, you guys are all done, you little mutants. I hope you guys had a good day and you got your DNA lab stations finished up. Go ahead and hand it in, and I will talk to you guys tomorrow, hoping the kids are better.